Hello and welcome to this Nature Research custom media webcast titled AAV Safety and Potency, Exploring the Analytical Maze of Gene Therapy. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by SVA Life Science. Gene therapy has the potential to revolutionize medicine. Adeno-associated virus technology is an essential platform for gene therapies as it has a low immunogenic profile and a diverse tropism for human tissues. However, a growing body of evidence suggests that the immune system can respond to AAV viral vectors used in gene therapies. This response is likely to be complex and multifactorial, just like the therapy itself. It's therefore essential to understand how the host immune response impacts the efficacy, long-term durability, safety, immunogenicity, and transgene expression potency in AAV gene therapy development. In this webcast, industry experts will discuss AAV efficacy and potency, the significance of and evaluation methods for humoral immune response, and address challenges and approaches for innate immune response. With us in the studio today, we have Dr. Abraham Scaria, who is the Chief Scientific Officer for Beacon Therapeutics. With 25 years of experience in gene therapy, he also serves on several committees for the American Society for Gene and Cell Therapy. Also with us is Nageswara Rao Kollu, who is the Senior Director at Apellis Pharmaceuticals. He's spearheading gene therapy programs to treat and potentially cure debilitating diseases. And finally, I'd like to introduce Boris Gorovitz, who is the Vice President, Non-Clinical and Clinical Bioanalysis at Regeneron Pharmaceuticals, and his main focus is on bioanalytical and PD support of diverse modalities of biotherapeutics. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to our speakers.
Hi, my name is Abraham Scaria, and currently I serve as the Chief Scientific Officer of a new gene therapy company called Beacon Therapeutics, where we are actively working on gene therapies for retinal diseases, both rare inherited genetic diseases and also some more prevalent diseases like macular degeneration. First of all, I want to thank the organizers of this webinar, namely SVA and Nature Research Custom Media, for giving me the opportunity to take part in this webinar and this discussion on AAV safety and potency, which is an area of great interest to the gene therapy community, as more and more AAV-based therapies are in human clinical trials and getting approved for use by the regulatory authorities. Since I'm the first speaker, I took the liberty of introducing some basic AAV slides. As you can see on this slide, AAV was discovered in 1965 as a contaminant in adenovirus preps. Here you can see the adenovirus capsids here and AAV, which is this tiny virus here. And turns out that over the last 50 years, we have found out that AAV is a depender virus and it cannot replicate on its own. So for replication, it requires co-infection with a helper virus like adenovirus or herpes virus. Now, wild-type AAV can infect humans. However, so far nobody has found any pathology or disease associated with AAV. Now, AAV is a very simple virus in a lot of ways. It has just two sets of proteins, the rep proteins and the cap proteins. The rep proteins are involved in replication of the genome and helps in packaging the genome into the AAV capsid. And the capsid is formed by VP1, VP2, and VP3 proteins. And this 4.7 kb genome is flanked on both sides by palindromic inverted terminal repeats. And these are involved in replication and packaging of the AAV. Now, as I mentioned, AAV can package 4.7 kb of genomic material, and currently it has become one of the most popular vectors for in vivo gene delivery, and this is mostly because of its safety profile. AAV, as you know, comes in various serotypes, and each of these serotypes have different tropisms and can infect different tissues in the body. And one of the examples is AAV2, which was the capsid used in Luxturna, where the target tissue was the RPE, or the retinal pigmented epithelial cells, in the back of the eye, all the way in the back of the retina. And this is now approved for patients with RPE 65 deficiency. AAV9 capsid was used in Zolgensma, which is a therapy that's been approved for SMA, and that is spinal muscular atrophy. And this is the reason for using AAV9 in this case, is that given systemically, AAV9 capsid seems to go through the blood-brain barrier and transduce motor neurons. And as you may know, AAV5 is also currently in clinical trials for several indications, and so is AAV-RH10. Now, at the same time as serotypes have been discovered, there's also been dozens of receptors and co-receptors being discovered for each of these AAV capsids. One example is AAV2 binding to heparan sulfate proteoglycan, and I'll tell you more about this in the next slides. Now, even though AAVs have been used in hundreds of clinical trials and look very safe and promising, there are certain aspects of the human immune response that still could pose some challenges. Now, here is a figure from a good review article written by Federico Mingozzi and co-workers, and it says that most humans have pre-existing antibodies which can neutralize the AAV and prevent it from binding its receptor and transducing the cell. AAV capsids can interact with toll receptor 2 on the cell surface, and this can trigger an innate immune response. Once the AAV is internalized and the single-stranded DNA is exposed, toll receptor 9 can be triggered, and this can also lead to an innate immune response. Once the capsid is encapsulated and once it's is destroyed, not only the capsid but also the therapeutic protein, in some cases it's a foreign protein, these can be processed by cellular machinery and presented through MHC molecules, either to generate a cytotoxic T cell response or an antibody response, or both in some cases. And as you know, the CTL response can lead to a reduction of transgene expression, and the antibody response can prevent re-administration of the AAV vector. So here is a list of observations both from preclinical studies and also from clinical trials from various tissue targeting. I mentioned both the innate and the adaptive immune responses. People have also noticed inflammation locally in the target tissue. A good example of this is the eye, where when you put high doses of AAV, you do see local inflammation. Hepatotoxicity has been observed in systemic gene deliveries, especially in the hemophilia trials and in the SMA trials. More recently, complement activation and thrombotic microangiopathy and also dorsal root ganglion toxicity has been observed when very high doses of AAV have been delivered systemically. One thing to remember is that every one of these observations is dose dependent, and it's also the route of administration dependent. So the key is to keep the dose as low as possible. You'll hear more about several of these things in the next two talks. I will focus the rest of my talk on retinal gene therapy. Now, there are two ways that are most popular in delivering genes to the retina. One is subretinal and one is intravitreal. And in the subretinal case, it requires a surgical approach where the vector is put all the way behind the retina by creating a subretinal bleb. This is a very highly efficient method to deliver genes to the RPE and the photoreceptors, the rods and the cones. However, this does require a very skilled surgeon to do this. The intravitreal approach is much easier to do. However, it is less efficient in getting genes to the photoreceptors or the RPE. But it is very good at transducing retinal ganglion cells, which are the first layer of the retina. And it is also a good way to deliver secreted transgene proteins into the eye. This does not require any fancy surgery. However, we have noticed that at high doses, you do see some inflammation, and more so with intravitreal than with subretinal. This next slide shows you the pattern of transduction by subretinal versus intravitreal, both in a mouse on the left-hand side and in monkeys on the right-hand side. And what you can see is with intravitreal administration in non-human primates, the most of the transduction is in the ganglion cells, and it's not uniform transduction. It seems like the ganglion cells just around the macula seem to get transduced the best. And also there's a lot of transduction in the front of the eye, in the ciliary body region. And of course, this is all done with wild-type AAV2 vectors. The same wild-type AAV2 vector, when you give subretinally, you transduce mostly retinal pigmented epithelial cells in the back and also some photoreceptors, not a whole lot of photoreceptors. So this is a study done by the Genzyme group and showing dose-dependent expression of a secreted protein, an anti-VEGF protein, after intravitreal delivery of wild-type AAV2 vector. And these doses are fairly low to moderate. It's only 2 times 10 to the 8th to 2 times 10 to the 10th. And the group shows that you can get persistent expression out to at least 6 months in this particular study, but they have shown in other studies that it can go out to several years. Also, what was noticed was at the highest dose, there was mild inflammation, and this was self-resolving, and it went away over time even without any treatment with steroids, and there was no damage to the retina even at the 2E10 dose. Now, as told you, everything is dose dependent, and there are a few ways that you can reduce the inflammation and improve the potency of your vector. And I'm describing 3 different methods here, but there are other methods also. One is to engineer a very potent therapeutic protein, so that then you need very little of the protein to get your benefits, so you go with less AAV vector. You can engineer the capsid to improve transduction of your AAV capsid for the tissue that you're interested in. The quality of the AAV vector is very important, and you need to reduce by purification all the impurities, and especially the empty capsids that don't contain any AAV genome. So I mentioned earlier how the Genzyme group was working with an anti-VEGF protein, and what they had done was engineered an extremely potent anti-VEGF protein. They did this by taking a extracellular domain of the VEGF receptor and fusing it to a human immunoglobulin IgG1, just the Fc part of that. And this created a very potent VEGF inhibitor, which worked at fairly low doses in a very stringent monkey model of laser-induced choroidal neovascularization. This is a classical model that people use for wet AMD disease modeling. What's shown here is that in the uninjected eye, when you place the laser burns, you get a lot of leakage of the fluorescein, whereas in the injected eye, which was injected in this particular case 6 months prior to this experiment, you can see that the laser-induced leakage is completely blocked by the SFLT01 protein, which I told you is a potent protein. And you can get away with very low doses of AAV2 here. And in terms of capsid engineering, Jennifer Sullivan and her co-workers have done a very simple experiment here. We know that AAV2 binds heparin sulfate proteoglycans, and what they've done is basically changed 2 amino acids on the capsid of AAV2, and this heparin binding knockout capsid now transduces subretinally very nicely all of the RPE and all of the photoreceptors, both the rods and the cones, as shown here in the mouse eye. And this data has been reproduced in monkey eye also. You can see the wild-type AAV2 barely transduces the RPE. This is an experiment here on the right done with a secreted protein where you can actually quantitate the amount of transgene protein you're making. And what's shown here is that the 2 amino acid changes can increase the subretinal transduction significantly, and the level of protein you can make is more than 1 log higher. Okay. So on the third topic of MT capsids, there have been several papers published now where people have shown that MT capsids can interfere with receptor binding and can reduce the transduction efficiency. And what's shown here in this last paper is a paper published by Adrian Timmers from AGTC in 2019. And what they've shown in this paper is that if you take a standard AAV prep, what they refer to here as the master prep, and injected IV at a dose of 1E11 vector genomes, you get inflammation both in the anterior chamber and in the vitreous chamber. And that's shown here with these black circles and this line here. Now, at the same time, if you take this master vector and purify it so that now it's enriched for full particles, you can see now that the inflammation is much lower both in the anterior chamber and in the vitreous, and at the same time you increase transduction as shown here by this green fluorescence protein staining. Now, even though it's enriched full, if you increase the dose of the vector to 3E11, now you lose some of that expression capability, and also as shown here at the high dose, some of the inflammation in the vitreous chamber comes right back up. So it's important to use low doses and just low enough to get the expression that you need. So just to summarize what I've just said, AAV vectors can lead to expression of proteins for very long periods of time in the eye. Gene therapies have shown proof of concept both in monkey studies and in human clinical trials. The inflammation and the other adverse events that are observed are at high doses, and if you can keep the dose low, you can get away from some of these inflammations. Local delivery in the eye and the CNS is preferable to keep the AAV dose low. And AAV manufacturing and purification methods to remove the empty capsids can improve both the safety and the efficacy.
And good afternoon. I am Nageshwara Kollu. I go by Nag. Currently, I am working at Epilis Pharmaceuticals and leading gene therapy programs. And today's my talk is targeting complement activation for enabling safer and more effective gene therapies. So, financial disclosure: I am employee of Epilis Pharmaceuticals, and these are some of the key points I would like to cover during this presentation. The first one is why the complex system is very complex and very hard to study. And second one is the key differences between the humans and mouse when it comes to the complement system. And third one is complement activation following the either AAV administration as well as the lipid nanoparticles, which people are using it for either vaccines or the gene editing programs. And the fourth one is the strategies we're building to mitigate the complement activation by using some of the complement inhibitors we have developed at Apellis Pharmaceuticals. So first of all, so I think many of you know complement cascade is an integral component of our innate immune response and a first line of defense against the bacterial infection. The system is composed of a number of plasma and a membrane protein and is tightly regulated. And the circulating complement components are produced in the liver, but it can also be produced by specific cells in other tissues. Complement activation can occur through three different pathways. The classical, which is the antibody mediated, alternate pathway, and lectin pathway. So here is the cool part of the complement system. The complement system isn't just fighting the infections. So it is like a multitasking superhero. Along with the fighting with the microbial infections, it also pitches into tidy up dead cells, which includes both the apoptotic cells as well as the necrotic cells. It also helps in healing the tissue, calm down the inflammation, and recently it has also found that some of the complement proteins can bind to the DNA and it can regulate the transcription. Based on the presence of the complement proteins, it can be divided into extracellular as well as the intracellular. When it comes to the extracellular, most of the complement proteins are produced in the liver, which circulate in the blood and through nearly every tissue of the body. Whereas the intracellular serves as the non-classical role, such as central regulator of the basal cell physiological process, which includes the mitochondrial respiration, glycolysis, autophagy, and gene transcription. So recently, several papers are coming up on the intracellular complement activation or complement pathway, and one of our colleague Martin Kolev, along with his postdoc mentor, working at several papers and showed intracellular complement could play a very important role in several cellular functions. In short, complement is just more than not a, just preventing the infections. It's a multitasking marvel, keeping the body in the top shape inside as well as the outside of the body. So based on this, because of the number of proteins involved in the complement system, and it present as both extracellular and intracellular, complement pathway can make it as a very complex system to study when it comes to the disease pathogenesis. Another thing is that like when you want to study the complement activation, so we need animal models. But the complexity with this complement system is that, as you can see, the complement system is partly somewhat conserved between the different animals. But when you look into the different proteins involved in the complement activation, there are significant differences was observed between the mouse and human beings. So these can be categorized into multiple ways. One, at the genetic level. So as you can see, there are several genes which are differ between the humans and mouse model. And the structure, so as you can see, C3 and factor B, even though they look similar, but there is significant differences in the amino acid sequence between these proteins, which includes the domains. And also the expression and distribution. So as you see in this slide, in the middle, C3 protein in the mouse models, which is around 0.3 to 0.4 mg per ml, whereas in human beings, which is 1 to 1.5 mg per ml, which is huge difference, around fivefold difference between the humans and mouse. And also there are some functional differences between the mouse complement proteins versus the human complement proteins, which makes very difficult very difficult to study these complement regulation or complement pathway activation in mouse models, and this will be very tricky to use them as a studying diseases which are linked to the complement dysregulation. So as you know, there are two vectors which are currently using for gene therapy vectors. These are the two vectors. One is the AAV and another one is the nanoparticles. So AAV vectors are basically using it for the replacement gene therapy vectors, whereas the nanoparticles people have used for the COVID vaccines, and also recently nanoparticles are significantly using for the gene editing applications. So as you know, AAV gene transfer holds a lot of promise for treating severe genetic diseases. But here is the catch. The immune system sometimes doesn't play nice with the AAV vectors, which can be a big hurdle for the effectiveness and safety of this therapy. Recent gene therapy trials using high doses of systemic AAVs have shown instances of complement activation within days to a couple of weeks after infusion. Elizabeth Krauss from Spark Therapeutics published a very nice review on complement activation. I encourage all you to go over that review to understand how AAVs can trigger the complement activation, particularly in the seropositive patients. In folks without antibodies against AAV, there is no change in the basal C3A levels after AAV stimulation. But in those with the antibodies, AAVs can kickstart complement activation, leading to increased C3A levels. This activation boosts the uptake of AAV into certain immune cells, which includes the dendritic cells, monocytes, as well as the neutrophils. And this activation boosts the resulted in the release of the cytokines, which leads to the inflammation. Now on to the mRNA lipid nanoformulations. These were primarily used for the COVID vaccines like Comirnaty as well as the Spikevax. These vaccines contain lower doses of PEG lipids but have been associated with side effects. So when you inject these lipid containing treatments, this can increase the adverse immune reaction by activating complement pathways, potentially causing the CARPA. The exact process behind the liposome-induced CARPA is still being unraveled, but the idea is that when complement gets activated, it releases anaphylatoxins such as the C5a as well as the C3a. These are the kind of troublemakers that kickstart a chain of reactions activating various immune cells and leading to the release of inflammatory cytokines as well as some of the other mediators, which resulted in the formation of the CARPA, which is a complement activation related pseudoallergy. So overall, either AAV or nanoparticles, which are shown to induce the complement activation. So here is the clinical evidence to show how AAVs or lipid nanoformulations carrying the PEG can increase the complement activation. So as you can see on the left-hand side in the table, many of the systemic gene therapies with AAV vectors are shown to increase the complement activation that has a severe adverse events in the patients. Along with that, the mRNA LNP code vaccines carrying the PEG lipids also shown to induce the complement activation in serum samples which are obtained from these patients. So overall, it is indicating that either AAV gene therapies or lipid nanoformulation related gene therapies could induce the complement activation that could lead to the severe adverse events in patients. So what we are doing at Appleis? So at Appleis, I think many of you know, we have developed a drug called pegcetacoplan. So this pegcetacoplan inhibits the C3, which is the upstream of C5, and has the potential to improve the control of complement activation. As compared to the C5, C3 can inhibit the both C3 split products within the C3A as well as the C3B, and also C5 split products, which is C5A as well as the C5B. So that means C3 has the more potential to reduce the complement activation as compared to the C5 based inhibitors. Overall, by targeting the C3 by using C3 inhibitors, you could reduce the endothelial activation, lysis, as well as the inflammation by using targeting the C3. So these are the different modalities we are building at Appleis Pharmaceuticals. So the first our approved product is pegcetacoplan, which is the inhibitor of C3, which is composed of 215 amino acid cyclic peptide conjugated to a linear polyethylene glycol molecule to increase the half-life. So this product was approved for treating both PNH, paroxysmal nocturnal hemoglobinuria, as well as another indication called GA, which is a geographic atrophy, a form of age-related macular degeneration. Along with this Pexataco plan, currently we are also building other strategies, which is siRNA, which we are trying to increase the durability because Pexataco plan for PNH patients, we need to give 3 to 4 times a month, whereas for the GA, these patients need to take every single time for every 2 months. So to increase the durability of these therapeutic strategies for preventing the complement activation as well as targeting C3, we are working on building siRNA. That means by using siRNA, we can treat patients 2 times a year. At the same time, we are also building a base editing approach. So instead of giving 2 times or around more than 2 times, we are trying to do a base editing approach so that you can do a one-in-done strategy for targeting the complement activation. So this is some of the data we have generated in collaboration with the Spark Therapeutics. So this was published in Immunology. This was done a couple of years back, and all the work was done by Eplis Pharmaceuticals, where we have provided our peptide, which is the pegcetacoplan, to target the C3 protein. So this is the data where it was shown that when you mix the patient blood, which are carrying the anti-AAV NAbs with NAbs and you add the AAV to this, it was shown that the patients who is carrying the more neutralizing antibodies, there was observed that the immune cells was uptaken by these AAVs, which includes the monocytic dendritic cells, monocytes, CDCs, PDCs. All these were where it was observed the increased uptake of AAV into these immune cells. Along with this uptake, it was also observed that increased amount of secretion of cytokines, which includes the IL-6, IL-1 beta, as well as the IL-8. But when we treat with our C3 inhibitors, which is the pegcetacoplan, it was shown that C3 inhibition could reduce the immune response to AAV in the blood, where it was shown a significant amount of reduction of the uptake of AAVs into these immune cells. All the cells we have tested, like monocytic dendritic cells, monocytes, or dendritic cells, where it was observed that by treating with our C3 inhibitor, which is APL9, we observed a significant decrease of the AAV uptake into these immune cells. Along with that, we also observed along with the immune cell uptake by the AAV, we also observed complement inhibition reduce the AAV-induced cytokine secretion in human blood. So as you can see in these graphs, following the treatment with our C3 inhibitor, we observed it was observed a significant reduction of the cytokines, which includes the IL-6, IL-1 beta, as well as the IL-8. We also did a transduction assays following the complement inhibition, and we observed that by treating with APL9, we observed a great amount of increased AAV transduction in vitro conditions. Overall, this indicates that by treating with complement inhibitors, we could reduce the AAV-mediated complement activation that resulted in the increased transduction in human cells. So this is the key takeaways from my presentations. And overall, our understanding of the complement system has evolved significantly since its description. Initially, it was considered as a pure innate immune pathway, but later it was realized that complement not only involved in the preventing the microbial infections, but it also involved in the several other functions. Another thing is complement. As you know, complement is very complex because pathway is composed of several proteins. Along with that, there are also significant differences between the human complement system versus the different other animal models. So that indicating that the studying of the complement is very tricky when you want to compare versus the animal models. So in the blood samples where Spark Therapeutics has shown that when you mix the NAb positive or NAb negative blood samples with AAV, it could induce the complement activation, particularly in the NAb positive cells. And also it's shown to increase the uptake of immune cells for the AAV particles. Along with that, it was also shown that following the AAV take of immune cells by AAV, resulted in increased amount of cytokine secretion. Interestingly, by treating with our C3 inhibitor, Spark Therapeutics has demonstrated it can significantly reduce the uptake of immune cells as well as the reduction in the cytokine secretion. So to conclude, we believe that inhibition of the complement system has potential to enhance many aspects of the gene therapy, which includes the increasing the safety, and also by increasing the safety and increasing the transduction, you can reduce the dose and ability to redose with the AAV gene therapies following the inhibition of the complement activation. With that, I will end my presentation, and I am happy to take if you have any questions. Thank you.
Hello, I'm Boris Gorovets, and first of all, I would like to thank organizers of the webinar for inviting me to present and share my opinions. And I want to thank you for attending and listening to this discussion. Next several minutes, I will give a high-level overview of the adaptive immunity-related questions as it is related to the AAV-based gene therapy modality. I will focus on the evidence and methods to assess pre-existing humoral immunity in particular. And as a quick note, the opinions expressed in this presentation are mine only. As a high-level message from this presentation, I would like you to recall that high and serotype-dependent pre-existing humoral immunity has been broadly reported by many groups. There are two main types of methods that are commonly applied to evaluate anti-AAV antibody presence, and these are total or TAB and neutralizing or NAB antibody detection protocols. Other than anti-AAV antibody responses should be considered during AAV clinical trial conduct, and I'll touch on that in next slides. The AAV-based gene therapy modality presents a unique case study of what I would argue is a clear case of high risk of induction of adaptive immune response when administered systemically. While probability of anti-AAV antibody induction after the treatment is very likely, the consequences of such response may vary and depend on many factors, including route of administration, viral dose, and others. The direct administration into immune-privileged tissues, such as ocular, has been generally viewed to generate low risk of antibody response induction and low probability of impact. Related, due to natural exposure to wild-type AAV virus, we should expect a high high prevalence of anti-AAV positivity in treatment-naive population. And in fact, high prevalence of total and neutralizing antibodies has been broadly reported. Detected anti-AAV humoral response is highly serotype dependent and can serve as a potential barrier to systemic delivery of the virus to the target cell, with a potential impact on the transduction efficiency. As reported by Dr. Wan and many others, a significant impact on expression of transgene was observed in various animal models. With this said, it is still unclear what level or titers of anti-AAV antibodies should be considered as impactful in clinic, and whether only patients with a negative antibody score should be admitted to an AAV-based treatment. In an excellent study published by Majovic et al., authors discuss outcome of AAV5-based therapeutic application for the treatment of hemophilia B condition. Patients were initially screened in a green fluorescent protein detection-based neutralizing antibody assay and admitted for the treatment based on the NAb negative status, with a later transition to a more sensitive luciferase reporter gene-based neutralizing antibody assay. It was discovered that some of the patients are in fact NAb positive and have a particular level of titer. It was reported that some of the patients with NAb positive status, as determined in the updated NAb method, still achieve therapeutic efficacy. Another interesting observation that we can make is how the correlation between TAB and NAb results in this specific study has improved after the transition from the GFP to luciferase reporter gene-based neutralizing antibody assay. And I will touch on this correlation a little more on the next slide. As reported by Long et al., we should expect a very robust and persistent humoral anti-AAV response after the treatment. And in the study reported by this group, high titers were observed in all treated patients for up to 3 years after the infusion. It should be also noted that no association was reported between the anti-AAV titers, post-dose, and transgene expression. Two types of methods, as I mentioned, are commonly applied to detect anti-AAV antibodies. To detect antibody neutralization activity, a cell-based protocol can be used and most often applied. And in this type of methods, antibody ability to impact viral vector transduction is evaluated typically and based on reporter gene signal. As a consequence, these assays are often referred to as transduction inhibition, or TI methods. It is crucial to appreciate that cellular transduction by viral vector is very complex process, and it has multiple steps, including initial binding and internalization of the virus, and then travel of the viral vector through the cellular compartments. And as a result, multiple factors may, in fact, impact efficiency of the AAV transduction, but only some of this related to the anti-AAV immunity. And the observed reduction in transduction efficiency may be, in fact, due to other factors, not antibody against the AAVs. And so Felice et al. Report that analysis of populational treatment-naive samples in TAB and NAB assays reveals presence of 4 sample groups, including those that are TAB and NAB negative, TAB and NAB positive, TAB positive and NAB negative. But what's most interesting is TAB negative and NAB positive. So this last group of samples suggests presence of other than immunoglobulin-based transduction inhibition factors. And I skipped, but I should add that the total antibody assays are typically ligand binding based and more or less straightforward and very common to what we have broadly used for protein biotherapeutic type of modalities. So together, this consideration suggests that NAb methods may overestimate anti-AAV positivity. The use of TAB protocol, total antibody protocols, has been favored recently due to the simplicity of the methods, ease of implementation, particularly if there's a plan to put in place a companion diagnostic method or similar. The overall TAB protocols present quite a solid alternative to the NAB or TI methods. I would like to conclude with a quick reference to potential induction of cellular anti-AAV immunity The CD8 positive T cell response has been reported by several investigators and was proposed as one of the potential mechanisms of elimination of AAV transduced liver cells, which will result in reduction of the transgene expression and impact on efficacy. And lastly, we should be aware and not forget about potential induction of anti-transgene product immunity. And here we should be able to employ many considerations that have been previously applied for protein-based therapeutics, including patient CRIM status or presence of non-human sequences. And at the same time, there's a very unique nature of transgene product expression, which is it happens in context of the AAV-based gene therapy, and will add some specific features to the anti-transgene immunogenicity potential. And lastly, I want to finish with these high-level messages and restate that high and serotype-dependent pre-existing humoral immunity to AAVs with potential impact on treatment, safety, and efficacy has been broadly reported. Specifics of the impact potential will greatly depend on many factors, including site of administration, AAV serotype, dose, and several others. While methods of detection vary, two main types of anti-AAV humoral response assessment are commonly applied, which are total and neutralizing antibody detection methods. And there's a shift towards use of total antibody protocols, as these are typically easier to implement and develop. They are also more robust and avoid detection of other than the antibody-based transduction inhibition factors. And lastly, a robust and sustained anti-AAV humoral response should be expected post-administration. With this, I would like to conclude. Thank you for your attention, and we'll open our webinar for the next presenter. Thank you.
Okay, so thank you very much to Boris, to Nag, and Abraham for those presentations. You can access some of the PDF articles that were referred to by going to the Resources tab in the auditorium, and you should be able to find those PDFs available. Okay, so I'm now going to move to our question and answer session. You can ask a question to any of our speakers here. To do so, please type it in where it says type your questions here and then press submit. Okay, so if I can ask you all to unmute and I will begin the questions. Our first question asks, why the low-dose enriched AAV is more efficient in transduction than the high-dose enriched version. Abraham, perhaps if I can ask you to answer that one.
Yeah, sure. Yeah. So this is, if you looked at that slide, besides the transduction, something else that happened was that the inflammation went back up at the high dose. So it's a combination of putting in more AAV capsids and more genomes. And like I mentioned, the DNA and the capsid can both trigger innate immune responses. So as you increase the load, you're going to get more innate responses and probably more CD8 responses too. In the eye, it's hard to measure these things because you can't get samples out of your retina. But that is my guess, that it's a combination of those things. So, and this is we see this even in systemic administrations. There is what we call a Goldilocks effect in gene therapy. At very low doses, you don't get anything, and then it goes up, and you hit an ideal level. And then if you keep pushing it, then things get worse. So that's probably what's happening in the eye also.
So if you keep pushing it, you get an equal and opposite response from the body, basically.
Yeah. So it's a risk benefit
Yeah.
thing.
Yeah.
You have to stop at a certain level with whatever benefit you're getting. Yeah.
Okay, thank you very much for that. Our next question asks, what are the side effects of inhibiting the complement system? Is it the case that it may have a beneficial effect on the therapy, but what if interfering on their physiological role by the inhibition, it actually causes some other medical condition? Nag, can I ask you to take that one, please? Oh, you just need to unmute. You can just give the My Connections tab a little wiggle and your unmute bar should pop up.
Yep, I can do it. Yeah, I'm happy to take that question. Yeah, it's a great question. So, but based on the clinical data from one program, which is our systemic approach, where it was shown that C3 inhibition is not a major concern regarding the safety. It is very much tolerable in those patients, particularly in the paroxysmal nocturnal hemoglobinuria patients, where those were treated systemically with the C3 inhibitor. However, for these kind of therapies for long-term complement inhibition, generally these patients will be administered vaccines to prevent any future microbial infections. But in case of the AAV gene therapies, we do not need to keep complement inhibition for long term. So generally, we might need to treat the patients maybe in advance around 7 days before treatment, and after maybe another 2 or 3 weeks might be sufficient to prevent the complement activation as well as increasing the or boosting the AAV transduction. So in that case, I'm not much worried on the any safety concerns following the C3 inhibition. That means it is very transient C3 inhibition, and yeah.
Okay, thank you very much. There's another question which I'd like you to answer, actually, please, Now, I think. It asks if there are any reliable in vitro or animal models to predict CD8 positive T cell mediated immune response in humans. And you spoke a little bit to the difficulties of translating between models. What would your response be?
Yeah, it's very hard. Like if you are, from my perspective, building an animal model for AAV gene therapies, it's very hard because, as you know, the immune system it's completely different from mouse to humans, and as well as the AAV response between the humans as well as between the mouse, it's completely different. So in that case, non-human primates might be a better opportunity in animal models. But again, and these non-human primates are healthy, and in that healthy environment, I'm really not sure the immune response between the AAV and immune response might be somewhat different as compared to the patients where the immune response to the AAV might be completely different. So in that case, it's very hard to recapitulate what is going on in human patients with the AAV response. In that case, like some of the people are using in vitro system like blood samples from patients and checking the immune response. So that might be the better way to understand the AAV immune response as compared to developing these animal models.
Okay, thank you very much. Moving on to our next question. This question asks about IgG proteases such as IDS or plasmapheresis, which has been discussed as potential tools to overcome immunogenicity. How doable will it be in the near future to re-administer this type of AAV vector? I think I will direct this to maybe to Abraham. Oh, you're on mute. If the toolbar is not coming up, just give the My Connections tab a little wiggle.
Yeah, there. Yeah. Yeah, this is about the enzymes that cleave antibodies. So, yeah, people have started testing those in preclinical models, and it looks like it does work to some extent. I think there's been papers from Spark Therapeutics and a few others. I think a group from North Carolina had published a paper. So it does work, and in terms of uh, translating this to the clinic, there are some enzymes that are in clinical trials now for mostly for transplantation purposes. I know there are some IDES type enzymes that are in clinical trials. So there is clinical data from humans that you can reduce the antibodies to whatever you're looking for. So maybe in the future we'll see this in clinical applications. Especially this is a big problem with systemic delivery. Um, if you're going subretinal with an AAV vector, it doesn't seem to be a problem, the pre-existing antibodies, but definitely in systemic applications. Yeah.
Okay, so perhaps I should ask Boris maybe if he'd like to also comment on this.
Yeah, I agree with Abraham, and I think there are many challenges, right? How do we re-administer the AAV virus, or we administer to patients that have high pre-existing titers, and what does it mean, high titers? So this is the $1 million question, or many more. If we can and re-administer, when can we re-administer? How to suppress immune response? And the use of the enzymes that cleave antibodies is one of the methods that have been investigated. There are challenges related to the nature of these enzymes, which are bacterial, of course, right? And they will bring their own immunogenicity potential into picture. So we need to not forget about that.
Okay, well, thank you very much for that. And I'm going to direct our next question to you, Boris. How do you evaluate the impact of pre-existing immunity to AAV vectors on the safety and efficacy of gene therapy treatments in clinical trials?
Right. So it's very much related to the question we just tackled.
Yeah.
And the question is, the answer is it's very hard, right? And the organization that is running a clinical trial is always facing this dilemma. Where do we draw the line of what patients do we include or exclude from the study? When it comes to local administration, for example, ocular, there's quite a bit of evidence that you may be able to include more patients, even if they have pre-existing antibodies, because it is presumed that the impact is reduced due to immune privileged location of that site. But in systemic administration, this is much more impactful. So how do you assess? Well, probably you need to move gradually, but it's a very hard decision. And I'm not aware of a solid evidence that is going to tell you the answer. There are many people who work on modeling of the impact, and still yet to see, what's the outcome of that effort.
Okay, and I think we just about have time for one final question, and it is again for you, Boris. In your perspective, how important is it to characterize the immune responses, both in terms of total antibodies as well as naturalizing antibodies?
Well, I think you may want to just pick your battle, right? So you either go after TAB or NAB. Yes, there are some organizations that have looked at both. I don't necessarily know how this collection of data may be used together, unless you're talking about research. So if you're talking about clinical development, I would just pick one. My preference would be to go with the TAB. It's just my opinion.
Okay, thank you very much. And on that note, we are going to have to finish for today. We are aware that there are a number of questions that we didn't manage to get to during the live session. And so we will get back to you after the broadcast finishes if you've provided us with your contact details. So I would like to thank Dr. Abraham Skaria, Dr. Nageswara Kollu, and Boris Gorovitz very much for their presentations today and for answering all of our questions. To all of you, thank you very much. I would also like to thank our webcast sponsor, Spara Life Science, and you, the audience, for tuning in to this webcast today. You will be redirected to a short survey. Please do take a moment to complete the questions. Remember, you can watch this webcast again at any time on demand at www.nature.com/natportwebcasts. Thanks for watching, and I hope you'll join us again soon.